Hello, thanks for joining us today for the latest Technology Networks webinar, Evaluating Microphysiological Systems for Drug Development. I'm Tom Koszczewski, the Director of Biology at CM Bio, and I'm here to introduce today's webinar. I'm pleased to have Dr. Alex Ribeiro joining us today as our presenter. A warm welcome to you, Alex. Alex has worked with the US Food and Drug Administration, the FDA's Division of Applied Regulatory Science since January 2017, and he currently leads the Integrated Cellular Systems Laboratory, where his research is focused on evaluating the use of human in vitro cellular systems for drug development applications. Alex received his PhD in biomedical engineering from Carnegie Mellon University in 2010, where he researched the mechanics of the nucleus of adult stem cells and cancer cells. He then became a postdoctoral fellow at the Stanford Microsystems Laboratory, where he worked to develop microfabricated devices that engineer biological properties of mammalian cells and analyze cellular mechanobiology. He later joined the Srivastava lab at the Gladstone Institute of Cardiovascular Disease in 2013, where he continued to study stem cell-derived cardiomyocytes within engineered microphysiological systems, leveraging novel tools to mature and functionally analyze these cells. At the FDA, Alex has been continuing his research in stem cell differentiated lineages and is also evaluating microphysiological systems as novel tools in drug development. Following this webinar, we will have a Q&A session, and we welcome any questions that you might have. You can submit any questions that you have for the Q&A session at any time during the presentation. To ask a question, you should enter your question into the box on the right-hand side of your screen and click Submit. We will answer as many as possible during the time available. For any questions we don't have time to get to, we will be sure to contact you offline with an answer. If you experience any technical issues, please click the chat icon in the top right corner to request further support. And please remember, you can ask questions at any time, even whilst watching the webinar on demand. So without further ado, I'll now hand over to Alex.
Thank you so much for the introduction, Tom, and thank you for the opportunity to present a general overview of the work being done at the FDA, and specifically in the Center for Drug Evaluation and Research. So, quick disclaimer, the opinions expressed in this presentation are mine, and they do not necessarily reflect the official views of the FDA, and I have no conflict of interest. Engineered cellular systems, microphysiological systems, these are all types of cell culture systems that were developed to enhance the physiology of cells in vitro and make cells have a function that resembles more the function of cells in vivo than the function of cells in traditional 2D culture systems. There are many terms that are used for these types of systems, microphysiological systems, organs on chip, cellular systems. In addition to calling these microphysiological systems, I'll also be referring to these as cellular microsystems today. Going back to the mission of our center, our FDA Center for Drug Evaluation and Research, our mission is to promote public health by helping to ensure that human drugs are safe and effective for their intended use, that they meet established quality standards, and that they're available to patients. As drug development tools, cellular systems have the potential to accelerate drug development and obviously reduce the associated costs with drug development. We can predict clinical results early in drug development. They can reduce and refine clinical testing, and they can be more predictive than current preclinical tools. In addition, they can be a solution for modeling rare diseases that is very difficult to do now with clinical trials. In our division of applied regulatory science, our goal is to move new science into the CDER review process and close the gap between scientific innovation and product review. So what do? We do two things. First thing is we perform mission-critical applied research to develop and evaluate tools, standards, and approaches to assess the safety, efficacy, quality, and performance of drugs. In addition, we perform expert regulatory review consultations in CDER to support the CDER regulatory effort. And we basically do critical review of existing knowledge, computational analysis if needed also in vitro and in vivo laboratory studies, and we also do translational analysis of preclinical studies, clinical trials, and post-market data. So when it comes to cellular systems or microphysiological systems, we evaluate these as emerging drug development tools. And as we're doing these evaluations and characterizations, we also support the CDER regulatory effort by providing expert opinion on any data or any kind of information that may be submitted with these systems in drug applications. So the scientific innovation that we evaluate in our lab are cellular microsystems. We're starting now to give an emphasis to the integration of induced pluripotent stem cells in these microsystems. So these induced pluripotent stem cells, they can be differentiated into any cell type in the body. And these cellular microsystems can provide a variety of different endpoints that then can be used in drug development applications to predict and detect human-specific drug effects. Because these systems have specific physiological properties and they do not replicate all of the regulatory components of organs or tissues, it's very important when using these systems to have organ-specific context of use. And I've noted kind of very general context of use, pharmacology, toxicity, mechanism of action, efficacy, safety, if it's new drugs or generic drugs. But this concept of context of use, I'm going to talk about it many times during our presentation today. And it's going to be central in characterizing and evaluating these systems. Before focusing on context of use, it's known in the field that lack of reproducibility is a main limitation in the field. And it's limiting the translation of these systems into drug development. And the reasons for this lack of reproducibility can be because of variation in device or fabrication, variation in device fabrication or handling, but also it can be because of variation in cell origin, isolation, and culture. So for fabrication and handling, the way systems are assembled, the way proteins are coded, priming, pumping, differences in media changes or operational calibration can cause lack of reproducibility or variability. When it comes to the cellular part of these systems, different isolation protocols, variations in genetic backgrounds, different types of media that are used, differentiation protocols if we're talking about iPSCs or even cell-specific handling, these can cause also variations in the results with these systems. So overall, in vitro systems are used early in drug development before clinical trials, and they have advantages relative to in vivo systems, mainly because they have the capacity to be more controllable for experimental conditions, and they can enable mechanistic understanding of data that may be difficult to perform at a similar level with animal tests or in clinical trials. However, for characterizing these systems, it's very important to start with clinical data, mainly because we do not believe that these systems are ready now to be used without a preliminary characterization of their context of use. And for this purpose, it's important to start with an hypothesis on mechanistic drug effects based on clinical data. And with this clinical data of specific compounds or specific drugs, establish context of use for cellular systems. And once these contexts of use are tested, then it may be possible to later use these systems that will be partially validated, or their context of use will be demonstrated, and it will be possible to use them later in drug development for specific applications. So for this, we need to develop validation strategies based on context of use. And most importantly, this is not something that a single stakeholder can do alone. This requires the collaboration between different drug development stakeholders. So as I mentioned earlier, it's very important to use compounds that represent specific contexts of use, compounds for characterization, and assays should represent these contexts of use. So for toxicity, for pharmacokinetics, for pharmacodynamics, and even for different applications such as efficacy, safety, and for new drugs and generic drugs, there's a history of different compounds that had different clinical results. So it's very important for different stakeholders to come together and define different applications, different contexts of use, define different compounds with specific results that can be used to represent these contexts of use, as well as with controls, and set up evaluation strategies for these specific applications. So key takeaways today on establishing strategies for characterization of microphysiological systems. As I've been mentioning, systems should be evaluated or developed for specific context of use. And I'm going to talk today about our work with the liver system, our work with the cardiac system, and also future systems that we're working on, and which are kidney and lung. Hopefully, I'll be able to convince you that these systems can answer focused mechanistic questions, and I'll give particular examples for this. I'll also mention that not just our center, but other labs in other FDA centers are also characterizing microphysiological systems, obviously for context of use that relate to the products that are regulated in those centers. iPSC differentiated cells are an opportunity for human and patient-specific evaluations. That's something that I'll also be mentioning. And I hope to, by the end of the presentation, to convince, as I've started doing in the previous slides, that standardization of performance is of key for enabling the use of microphysiological systems, because this gives them reliability. Multiple stakeholders are necessary to translate new methods. So this is a general slide from my friend Brian Burridge at the National Toxicology Program and National Institute of Environmental Health Sciences. And as it is demonstrated in this slide, in order to go from analytical validation that can be done in an academic lab or in a research facility lab into translational qualification, there are different key enablers that need to be followed. So our lab, as I've mentioned, we complement work done by other researchers. We don't develop new systems, we don't fabricate new systems. And because of this, we collaborate a lot with different stakeholders. And our contribution to this effort is in replicating biology, demonstrating pharmacology and toxicology, and testing for analytical reproducibility and doing comparative studies. However, there's much more that needs to be done after this. And for this, it's necessary to have consortia, public-private partnerships, basically mechanisms that enable the participation of different drug development stakeholders. And we collaborate with these. So just giving an overall overview of the history of FDA's involvement with microphysiological systems, it started very early in 2010 when the concept of organs on chip and microphysiological systems started happening. And we were very much involved since then with NIH, with DARPA, with NCATS. And recently in 2017, we started signing agreements between FDA labs and developers of these systems to evaluate these systems in our labs. And this year, additional agreements have been established, and we anticipate that more agreements will be established for evaluating these systems in different contexts of use. So overall, a lesson that we've learned is that our participation, either as advisors or as active researchers, have been critical to the development of this technology. This slide basically shows a little bit of all the efforts that are being developed at the FDA in this field. As I've mentioned, In the Center for Drug Evaluation Research, we're evaluating a liver system right now from CN Bioinnovations. CBER, Center for Biologics Evaluation and Research, they're working with a Korean company, CryoChips. They're also developing their own systems for specific applications related to biological products. The National Center for Toxicological Research, they have a collaboration with Tissues, which is a European company also. In addition to our collaborative agreement with CN Bioinnovations, we're also collaborating with the Healy lab in Berkeley with the use of iPSC-based cells for microphysiological systems. And CIFSEN, Center for Food Safety and Applied Nutrition, they have an agreement with the company Emulate. We in CDER initiated our focus on hepatic and cardiac systems because they account for over 75% of safety-related withdrawals. And liabilities often are detected often late in drug development. It's also acknowledged in the field that there's a pressing need for more predictive in vitro tools. In fact, there's this study that shows that a 10% of improvement in predicting failure before the initiation of clinical trials could save up to $100 million in the costs associated with drug development. In addition, overall, modeling toxicity in the liver is of high importance for drug development, since most drugs are metabolized in the liver. A lot of liabilities happen because of drug transport problems. There's lots of drug-drug interaction questions that are of interest for drug development. And there's an overall need for physiologically based pharmacokinetic and pharmacodynamic models when it comes to data in drug clearance, in drug-drug interaction, metabolism, etc. So when we started talking with developers, we realized that for developers, this is how they see microphysiological systems in general. Here we're talking about heart and liver, but they basically see these systems to have bioengineered factors that are combined in microsystems and cells that are seeded or loaded into these microsystems and then cultured. But for us, we actually see these systems as a tool that can be used for drug development, where we incubate drugs and then we look at specific function, metabolites, biomarkers, phenotypes that are relevant for a specific application. These applications, as mentioned, can be toxicity, absorption, distribution, pharmacokinetics, pharmacodynamics. But for regulatory use, these systems must operate robustly, originate reproducible results, and definitely improve on gold standard, 2D or others. So for this purpose, we establish criteria for our initial evaluations that is completely based on this need, the need for robustness and reproducibility. We tend to work with systems and cells that have been published, preferentially used in different laboratories. And because we want to evaluate something that can be used universally, we tend to favor systems that have been demonstrated with commercially available cells and cells that have quality control protocols to ensure that we don't work with something that only us or a few labs can work with and other folks cannot work with. So based on this, the general criteria for evaluation of cellular microsystems in our division is to first look at site-to-site variability in terms of producing results. And this is where we work with system developers that repeat our experiments, or we repeat their experiments. We also evaluate chip-to-chip variability. So for the same experiments, we use different chips, chip batches with different cell donors, and see how that affects results. And for this purpose, we focus on PTMS protocols, where preparation of devices and cells, treatment with drugs, and measurement schedules are well detailed and controlled as much as possible. So this slide kind of shows overall the systems that we are characterizing in our lab. So the first one is the CN Bioinnovations LiverChip that gives different outputs in cell death, metabolism, biomarkers, transport, and gene expression, among others. We're also looking at the engineered heart tissue, the EHT, that gives outputs in contractility, calcium cycling, duration of contractions, and this one we use iPSC-derived cells, while the CN Bioinnovations so far we've used primary hepatocytes and primary Kupffer cells. And to use iPSC-derived cells, we've been also collaborating with Berkeley in their heart-liver system to better understand how to integrate iPSC-derived cells in microphysiological systems. So as you can see, we kind of have a repetition. So we have a liver chip and we have a liver system in Berkeley. We have an engineered heart tissue and we have a heart system also in Berkeley. So the purpose of evaluating several systems that can model the same organ is also to understand how different the performance of distinct systems can be. To determine commonalities between systems, this is important to develop standards criteria that then can lead us to recommend quality control assays that will definitely benefit the drug development field. And our goal right now is to initiate the development of context of use for different systems that we're using and also work with other stakeholders in doing the same thing with other systems. So today, I'm going to talk until the end of this presentation about some data and some work that we've been doing with CN Bioinnovations LiverChip, and then I'll just mention a little bit of the work that we've been doing with the engineered heart tissue, EHT, as well as the Berkeley system. So it's known that the liver microenvironment is three-dimensional. The cells are exposed to fluid flow, and it's also multicellular. LiverChip has been demonstrated to have long-lasting hepatic properties, which we've reproduced in our lab. It enables the possibility of culturing heterogeneity of cell types in the same system, so it enables co-cultures. And it seems to be universally reliable and robust properties based on the fact that it's commercially available, and different folks, either in academia and drug development industry, have published papers with this system. So this is how it works. So basically, in this, the right low corner, we can see the system is of the size of a 6-well plate or 12-well plate. It has 12 wells, and each well has basically a microfluidic chip. And this microfluidic chip enables the media to flow through this scaffold. And this scaffold contains cells that are organized in 3D. So basically what we have here is 3D culture, co-culture with cells under fluid flow. This is basically the microenvironment that differentiates cells that are cultured here from cells that are cultured in other systems, 2D or spheroids. When using multiple cell types, we need to think about how does this improve the ability to predict different mechanisms. And then we think about toxicity as being one of the applications. Here in this slide are summarized some of the mechanisms of liver toxicity that can be induced by drugs. If we look at necrosis and apoptosis, it can be discussed that these are hepatocyte-centric phenotypes. But when we look at inflammation, Kupffer cells are definitely important to be included when evaluating the toxicity of drugs that involves inflammatory factors. For immune response, it's currently not possible right now to emulate the complexity of the immune system, but there are different folks, both in academia and also with developers that are working on this, and we're looking forward to seeing how the field is evolving. I'm just going to give a specific example of a study that we did in our lab where we looked at co-cultures of hepatocytes and Kupffer cells to look at drug effects that are toxic and involve an inflammatory setting. Trovafloxacin is a FDA-approved drug. Trovafloxacin is a very similar drug to levofloxacin in terms of chemical structure, but it has restrictions for use due to hepatotoxicity. It's not safe. So we developed a protocol for testing these drugs with LiverChip, where we define treatment schedules. We co-dosed with LPS, lipopolysaccharides, to induce inflammatory signaling, and we used 3 different concentrations. In this slide, you can see that the LiverChip is able to detect Trovafloxacin toxicity. It's a very busy slide, so on the left side we have LDH, which is a very well-established marker of cell death. And we can see that in the presence of LPS at 100 micromolars, there is cell death. On the top slide, we see experiments that were done in site A, which was the site of the developers. And on the bottom 2 graphics, we can see experiments that were done in site B, which was the FDA labs. And we see concordance in data, not only in cell death, as I mentioned before, where in the presence of an inflammatory signal, we see cell death, but also in the decreased activity of CYP3A4. This decrease in CYP3A4 is also observed when we add LPS, but we don't see an increase in LDH, meaning that despite a decrease in metabolic function, there's no cytotoxicity. So this slide shows that the same experiment done in different sites with different batches of Kupffer cells can result in similar data, therefore, suggesting that this system is reproducible, at least for this specific context of use. In this experiment, we use the same batch of hepatocytes. We use the same drug catalog numbers. We used LiverChip devices and instrumentation that were also the same. What was different in this experiment between sites? We used different analytical instruments, so different plate readers, plate shakers, and the lot numbers of the assays, despite using the same assays, were also different. So for CYP3A4, we also did albumin and for LDH. So the next slide addresses how the LiverChip function lasts longer than sandwich cultures and spheroids, and the magnitude of function is higher than that the function in sandwich cultures and spheroids with the same cells. So we used primary hepatocytes, and we basically assayed albumin throughout 20 days after seeding these cells. And as we can see here, the production, and this is normalized production to number of cells and amount of media, the production of albumin is much higher in the LiverChip than it is in sandwich cultures or spheroids. This was done with a specific batch of hepatocytes, primary hepatocytes, and now we're basically doing with another batch to see if we can replicate these results with another batch, and we'll be publishing those results soon. The key takeaways for the LiverChip is that 3D culture and media flow enhances and prolongs activity of hepatocytes. These systems must be developed for mechanistic evaluation of drugs, as seen specifically for the role of inflammatory factors. And the use must be standardized to ensure reproducibility. And this is work that we will be doing with further toxicants, in addition to the work that we've done already with the developers with levofloxacin and Trovafloxacin. So before transitioning to the work that we're doing with cardiac systems, I'll just give a note on the use of human cells. These can be primary or differentiated from induced pluripotent stem cells. So primary cells are basically isolated from either tissue biopsies or from human patients, and induced pluripotent stem cells are reprogrammed from somatic cells and differentiated into the desired human cell type. As noted for the LiverChip work that we've done so far, we've only used primary hepatocytes and primary human Kupffer cells. The next work that we are planning to do uses iPS hepatocytes. For cardiac applications, we have solely used iPSC-derived cardiomyocytes. And the reason why this happens is because, as demonstrated, it's possible to get primary liver cells from donors. And these cells can be frozen, they can be thawed, they can be cultured, QC'd. It's something that has some, there are some difficulties associated with all of these, but it's possible to do it. However, with heart cells, cardiomyocytes, it's very difficult to maintain these for a long time, and it's also very difficult to access these. And to my knowledge, these cells that when they're frozen, they basically lose their properties and they cannot be thawed. So that's why iPSC-derived cells, even if they have some problems in terms of maturity, they are without question the only available alternative to include in microphysiological systems. So the preliminary work that we did with these cells was to connect them to the liver chip. So we basically had cultures of iPS cardiomyocytes. We added drugs to the liver chip, and then we added metabolites of these drugs to the 2D cultures of iPS cardiomyocytes. And we did this even before thinking about evaluating heart microphysiological systems or heart microsystems. And I'm now going to show you an experiment that we did to demonstrate this capability of metabolizing something first in the liver that then is added to a cardiac system. So we use terfenadine as an example. So as shown in this slide, terfenadine and its main metabolite. Terfenadine is cardiotoxic, metabolite is safe. Terfenadine is cardiotoxic because it blocks potassium channels. This induces a prolongation of action potential duration, and the clinical outcome is basically ventricular tachycardia. And this only happens if it's not metabolized. So, if patients have a good metabolism, probably they will not suffer a cardiotoxic event. So the experiment we did, preliminary experiment, was to incubate 5 micromolars of terfenadine in the liver chip for 2 days. And then we basically added this metabolized drug into the cardiomyocytes cultured as a monolayer, as I mentioned earlier, and we looked at calcium as an endpoint, calcium flow or calcium cycling. The specific phenotype that we looked in terms of calcium cycling was the time of contraction. So it's known that the duration of calcium cycling is a proxy to the duration of the action potential. This is what we looked at. And this is the data that we got. So when we add terfenadine alone to the cardiomyocytes, we see an increase of the contraction time relative to control, which is the mechanism that is known to occur when terfenadine is added to these cells. However, when the drug is metabolized in the liver chip, we don't see that increase, and therefore we think that it is safe after being metabolized as expected. So just a quick note, no cytotoxicity was observed in this experiment in the liver chip or cardiomyocytes. We tested for that. So where are we going now? In addition to testing more drugs, which is something that we're currently doing, we're also starting a collaboration with the Healy lab from the University of California, Berkeley. They have developed heart chips that induced alignment of iPS cardiomyocytes in 3D and enables functional analysis. And this is very well published. It was published in 2015. As you can see in this slide, these systems try to emulate the dimensions of the human ventricle. They align cells and align the contractile structure, and it's possible to get contractile readouts from these systems, as well as other readouts, such as action potential duration and calcium flow. Very similar to what I just showed. In addition, they have developed a liver chip that was designed to culture iPSC hepatocytes, and this work has been published recently on bioRxiv, so it's still very early. And they have the ability to connect the LiverChip to the cardiac system in a controlled manner where media can flow and the flow of media can be controlled and in terms of transitioning from one system to the other. So why is it important to align cells in this way and to have them in 3D? Biggest problem is that for iPSC-derived cardiomyocytes, if they're not aligned and if they're not in 3D, they have fetal-like properties. They do not resemble at all the morphology and function of primary cardiomyocytes. They have fetal-like properties. However, I don't think, and the field doesn't think that this is a reason to give up on these cells because, they do have human-specific properties. They do last long in culture. They easily attach to cell culture platforms in opposition to, primary cardiomyocytes. And most importantly, they can be genetically edited to emulate or represent specific patient-specific disorders or population-specific properties. So to take advantage of the potential of these cells, different folks in the field have been developing engineered cellular systems where they recreate the microenvironment of the heart with biochemical cues, with biophysical cues, and with bioelectrical cues. The engineered heart tissue that I'm showing in this video is a good example of this effort. There's many other systems that have been developed with similar capabilities or even with improved capabilities. Engineered heart tissue is the most published one, and it's the reason why we kind of started working with it. And it was initially developed only with the purpose of measuring mechanical output, the contractility of iPS cardiomyocytes, because single cells, when they're beating, it's not really possible to measure forces. However, as the system started being characterized, the developer started realizing that it was an ideal bioreactor to mature the function of these cells, with where it's possible to combine mechanical strain, chemical compounds, different microenvironmental cues that further enhance the maturity of these cells. We've started working with these systems in our division. We have an engineer heart tissue testing system, and we are able to develop these engineered heart tissues. And so far, our main concern was to get reproducibility with the results that have been published by the developers, as well as reproducibility between batches of engineered heart tissues. So in this slide, what you can see is different parameters that can be attained from the contractile analysis of these engineered heart tissues. And as you can see two things. You can see that, one, there's a lot of reproducibility between tissues. Two, these systems change with time. And it is known that after day 15 or after day 21, they kind of stabilize and become stable for like weeks, which is the time when we are planning to do drug experiments. So the tissues are performing in our hands as published. They last long in culture, and the work in progress that we're doing right now is basically in drug testing, and it's something that we hope to publish soon. So the key takeaways for cardiac systems is that iPS cardiomyocytes are a viable solution for using in microphysiological systems. They do have fetal-like properties, and because of this, 3D aligned microenvironments are used to enhance functional maturity. Connecting cardiac to liver systems allows for studying the effects of metabolites, as demonstrated in our preliminary data with Terfenadine. So before wrapping up this presentation, the future work, we're transitioning also to other systems and other types of applications. We're very interested on evaluating kidney on a chip systems or kidney organoids to look at outputs in absorption, clearance, accumulation, and metabolism. We want to do the same thing with the lung on a chip, and we're starting now to develop an agreement with CN Bioinnovations to also use this system in our labs. And something that we're also looking forward to do in the iPSC-derived field is to differentiate cells from patients that have differences in pharmacokinetics between each other and see if we can emulate these differences in pharmacokinetics between patients while using cells derived from patients in microphysiological systems. This is kind of where the field is going, in addition to continuing our evaluation of liver systems and cardiac systems. So just a reminder of the key takeaways. from today on establishing strategies for characterization of microphysiological systems. We believe that systems should be evaluated or developed to be used for specific context of use. And I hope I was able to show more or less where things are going, the field is going in terms of work with liver systems, cardiac systems. They should answer focused mechanistic questions for this purpose. There's different labs in the FDA that are characterizing microphysiological systems for specific context of use that relate to the products that are regulated in these centers. iPSC differentiated cells are definitely an awesome opportunity for human and patient-specific evaluations, but there's still a lot of questions to answer when it comes to their maturity. But most importantly for the field, standardization of performance is key for enabling the use of microphysiological systems in drug development. And that's something that we've been focusing so much in the beginning, because without this, we cannot move forward in setting context of use. So I would like to acknowledge all our collaborators at FDA, as well as our external collaborators from DARPA, CN Bioinnovations, and the University of California. In addition, I would like to acknowledge the work of researchers in the lab, postdoctoral fellows, biological scientists, summer fellows, as well as former trainees that were involved in this project. And at this point, I would like to open the floor to any questions and discussion if we have time for that. Thank you so much.
Thank you very much, Alex. I'm sure you will all agree that that was a really great presentation. So now we'll begin the Q&A session. So just as a reminder, to submit a question to Alex, you should enter the question into the box on the right-hand side of your screen and click send. We will answer as many questions as possible during the time available, but for any questions we don't have time to get to, we will be sure you are answered offline as soon as possible. So we've had a few questions submitted already, so I'll move straight on to these. So Alex, our first question is, to you, what are the criteria required for a great, robust and reliable organ-on-chip model?
That's a good question, right? Obviously, depends a lot on the organ system. So the specific biological properties that define that system depend on the organ and cells that are used. Functional stability, a system that is not functionally stable most likely will not be reliable for using. Long lasting, not just being stable, but having a function that lasts for a long time. In relation to the origin of cells to be used, having cells that independently of the cells that are used, it's functionally stable, and this is all important to basically yield reproducible results. So I think it's important to have a set of compounds that define the function of a specific organ system, and if adding these compounds at a specific concentration yields reproducible results all the time that is done, then I think all of these combined show that we may have a robust and reliable model.
Thank you, Alex. So our next question is, are all the cells used human in origin, or are you also examining chips with animal cells to allow closer comparison with the in vivo data?
This is a very good question. A lot of, mainly for toxicity studies, off the top of my mind, I'm thinking about developmental and reproductive toxicity. Basically, animal models are used for in the preclinical testing of compounds and of this type of toxicity. So it does make sense to use animal cells and compare the results of chips with these animal cells to the results of all the tests that have been done, for example, for this specific context, the developmental and reproductive toxicity. There are other contexts in terms of toxicity that also use animal systems. So it does make sense to start evaluating how these systems perform in relation to the animal models that are commonly used now for testing different types of toxicity. we haven't yet done any work with animal cells, just because we had to prioritize on the use of human cells. But it's definitely something to consider, and it's definitely a need in the field.
Thank you, Alex. Our next question is, what do you think the future of organ-on-chip technology will be in terms of regulations for preclinical drug testing? Do you expect this to be a requirement before entering the clinic?
Well, it's difficult for me to predict if it will ever be a requirement. I think what is a requirement in terms of making decisions during drug development is to do good science, to have good data, and to have data that folks that are evaluating that data have confidence in it. So I think as long as organs on chip, microphysiological systems, whatever technology, as long as it demonstrates to be predictive, I think then there's no reason not to use it, even without any requirements. As long as science is good, I think that's what everybody wants when evaluating drugs, is to be sure that the results of the assays are predictive and can reliably help make decisions if the drug is safe, if the drug is efficacious or not.
Absolutely, thank you. So our next question is, there are now commercially available organ-on-chip models for a range of organs. Are there any organs or models that haven't been created that you see a great need for in drug development?
Well, there's been a lot of models that have been created so far, and the field has been evolving really fast in the last 10 years. I think it's kind of something that I've been mentioning the whole presentation is the context of use. So instead of thinking about specific organ systems, I can answer the question to specific context of use if that would satisfy. My colleagues commonly ask for context of use, for example, related to developmental and reproductive toxicity. The guidance for that type of toxicity, the FDA guidance was just published recently, and I think for the first time, I hope I'm not wrong, they did mention the possibility of using alternative systems to animals, not just in vivo but also in silico. So there is definitely a need for context of use in developmental and reproductive toxicity. These systems could also be developed or perfected for drug-drug interactions, to look at juvenile toxicity, neonatal toxicology, something that several of my colleagues are very interested also, as well as myself, is to what extent can the immune system be recreated in these systems. There's been done a great job has been done in terms of recreating inflammatory factors, and there's very good data published on that. But there's other components of the immune system that have not been modeled yet, and to what extent can these be modeled? I think at least for the interests of the folks that I work with at the FDA, this is kind of where our interests are.
Thank you. Our next question is, as far as lung on chip, do you consider the culture system under air-liquid interface?
That's a good point. Yeah, it really depends on the context of use. For the problem of working with air gases is that it's often challenging to contain compounds in gas phase or within a chamber, because gas leaks very easily, and normally we need to add a lot of complexity in terms of pressurized, all that stuff. So it's always challenging to work with air when it comes to handling compounds. But then there's the other side, which is the physiology of the system, right? So different publications have been shown that the air-liquid interface, or inducing an air-liquid interface, seems to enhance the physiology of the function of epithelial lung cells. So if we think about that, it does seem to be important to recreate the air-liquid interface. But handling compounds as gases instead of liquids, instead of in solution, can be very challenging.
Thank you. So how are the prospects of using this system for therapeutic gene delivery?
Good point. That's something that folks from CDER would probably answer more, but or answer better. On my side, because at least in our hands, the systems that we have tested, their function lasts for a long time. Normally, these gene delivery take a while to happen. The gene delivering processes can take up to days, and also the cells take a while to recover from these. So I think, given the fact that these systems last long in culture or longer than the traditional systems, at least there's the advantage of having enough time for testing gene delivery strategies. But in terms of kind of the regulatory applications, I would follow up with folks from the Center for Biologic Evaluation and Research in the FDA.
Thank you, Alex. I know we have got quite a few questions coming, but unfortunately that's all we've got time for today. But just to remind everybody, any questions that we didn't have time to get to will be answered offline as soon as possible. You can also continue to ask questions even when watching the webinar on demand. So shortly the webinar will be made available on demand. So if you do have any friends or colleagues that you think may be interested in the webinar, please feel free to share the link. So thank you all for your time, and thank you, Alex, for a great presentation.
Thank you.